Okay, so we're going to talk a little bit about protists today. My goal is for us to finish up 21.2 in this video. So we left off discussing how protists were a very diverse collection of lineages, and we discussed how you can find protists in different aspects, whether they're singular or in a colony. Well, today I want to start off by talking a little bit about the cell structure of a protist. So protists are eukaryotes, which means that they all have a nucleus, and most of them have eukaryotic organelles. We can find things like the endoplasmic reticulum in them, and also the Golgi bodies. Most single-celled protists have a protective layer at their cell surface, such as a cell wall. It could be a hard mineral shell, or it could be a flexible pellicle, which we'll talk about. So these are some of the protists, so like green algae and dinoflagellates. They have that cellulose-containing cell wall. Then radiolera and diatoms, they have a glassy silica shell. And the euglena, this has the flexible pellicle, which is just basically a plasma membrane that has elastic proteins just under it. So depending on the protist depends on what type of structure that it may or may not have for locomotion. And protists use a wide variety of structures, such as the cilia. We also see flagella. And then sometimes we see these cytoplasmic extensions that are called pseudopods. And we know that pseudo, anytime that we see that, pseudo, of course, means not real or fake. And so this case, this is something that is made within the protist cytoplasm that allows them to kind of reach out and move themselves. Some protists have vacuoles that they will basically suck in a lot of water and then squeeze down, expelling that water, and it allows them to kind of shoot forward. The best example I could give you would be like air in a balloon, and when you let go of that balloon, how the balloon, goes forward and kind of goes all over the place. Well, this is the same thing, but it's water that's allowing it to do that. Euglena have a contractile vacuole that allows them to suck up that water and then expel that excess water through a pore. Many protists have chloroplast. So cyanobacteria evolved through primary endosymbiosis. And a common ancestor of red algae and green algae, and then later green algae and red algae evolved into chloroplasts and other protists by secondary endosymbiosis. And then some mobile photosynthetic protists have an eye spot, and this eye spot allows them to detect light, and then that allows them to move to that particular area. And so I wanted to show you this picture right here. So this is the primary endosymbiosis. Of course, we know symbiotic relationships are typically between two organisms. But in this case, what happened was you had this heterotrophic protist, and it took in cyanobacteria through this endosymbiosis. And so basically it just took it in. And now this protist can make food because it has a chloroplast. And we see this in red algae or in green algae. Then later, Later, another heterotrophic protist was able to engulf this algae right here, and that's what allowed it to create this chloroplast with these four membranes that we find around it. So most protists are obligate aerobes. But some are anaerobic protists, or some anaerobic protists have modified their mitochondria so that they don't use oxygen as that final electron acceptor. For those of you who had dual credit biology 1, when we studied cellular respiration, aerobic cellular respiration, at the very end of the cycle, when we went through the electron transport chain, the hydrogens attached to oxygen that, of course, was taken in through respiration, and it formed water. Well, in this case, these anaerobic protists have learned how to modify their mitochondria so that they don't necessarily need that oxygen to pick up that final hydrogen there. Protists include autotrophs and heterotrophs. Some heterotrophic protists live in the soil, and they will feed on decaying organic material or bacteria. Other heterotrophic protists live inside larger organisms as a parasite, and this is, or in some other cases of symbiosis, but a lot of times as a parasite. And then, of course, autotrophic protists, they have chloroplasts, and that allows them to, of course, go through photosynthesis, carry out their own photosynthesis. So let's go through a little bit of plankton and talk a little bit about that, and then we'll move on from there. So the majority of protists are free-living and aquatic, and they form something known as plankton, these little tiny drifting swimming organisms that we find in lakes and seas. Phytoplankton is photosynthetic organisms, or are photosynthetic organisms, and then zooplankton, they are heterotrophic organisms. So, of course, we know autotrophic means that they, of course, can make their own food. Heterotrophic means that they have to look for their nutrition elsewhere. And so, you obviously, a lot of diversity in metabolism. Because you look just right there, autotrophs, heterotrophs, and we even have some that are mixotrophs, which basically are photosynthetic, or they can be heterotrophic, just depending on the conditions that they are in. So I will say that when we talk about protists, we have to realize that they have been around for a very long time, and they have learned to adapt to the conditions that they come in contact with, or that they are find themselves meeting, and be able to adapt and change so that they can survive, because that's what evolution is. It's that survival of the fittest. So, just something to think about. These are pretty tough little organisms. Some of these protists will live in moist soil. Some of them live inside of organisms, and just depending on what they're doing, they could be beneficial to the environment or to their host, or they may be the only ones that are benefiting. So most protists reproduce asexually when conditions favor growth. So when they need to grow, when the conditions around them are optimal, then they will go through asexual reproduction. But they can switch, or will switch, to sexual reproduction when the conditions do not favor growth. So they're always looking for a way to go through reproduction, to continue to grow, to continue to reproduce. For protists, the dominant form of the cell that we see through most of the protist life cycle is the haploid cell. Only the zygote is a diploid. We see that in Plasmodium, which we will talk about later on. Some protists will demonstrate what's known as alternation of generations, which is basically where they will go from haploid to diploid states. Algae does this. It will go through this alternation of generations, cycling between this haploid and diploid state. So let me show you how this works. So right here you'll see the haploid dominant cycle. So the majority of the time it's spent as a haploid. And then right here you'll see that this is a diploid dominant cycle. Majority of the time is diploid. And then this one right here, this is where it will alternate generations. So basically the zygote will go through mitosis, form this multi-celled body, and then it switches to meiosis, forms spores, and then keeps on going. So it's around and around and around and around like this. So basically it's kind of to me, it's almost like a combination of this is what you're looking at. And that's going to be that alternation of generations. So at this point, I would suggest that you go and read 21.2 pretty closely. And I would, as I was reading through it, I would look at my notes that you've been filling in. And if you have questions, just drop that question in the Google Classroom. You can do it on the stream, and I'll try to explain it to you a little bit better, so that way we can make sure that you understand how protists are so diverse, how they metabolize, and how they reproduce. So I look forward to talking to you next time about the different types of protists, and we'll be looking at excavates in the next lecture.